So now that we understand how DNA is replicated, the method anyway, let's take a look at who's involved in copying your DNA. So the steps that we're going to take, and then the participants, the enzymes that are involved. So if we want to replicate DNA, the first thing we have to do is break it into pieces because it's too long. We've got to unwind those pieces, exposing the bases. We have to replicate or copy those strands, and then we have to put it all back together. And all of that is done by enzymes. Remember, enzymes are proteins that have very special functions. They're catalysts. They speed up these chemical reactions. They allow them to happen, and they do that by holding molecules and pieces in the right orientation to one another. So the first thing we call this first step initiation, to start, right? Initiate means to start. So we're going to start. And there's an enzyme called helicase. Now I want to talk a little bit about enzyme words, enzyme names, because you can always tell that an enzyme is there when you see the ending ASE. ASE is the flag that means this thing is an enzyme. Okay, so anytime you see ASE. And then the front part of the name, the old way of naming them, was to tell you a little bit about the molecule that the enzyme acts on, called its substrate, or what it does. In this case, helicase sounds like helix. And so helicase is an enzyme that works on the helix. And in fact, what it does is it zips open the helix. It opens it up. It's a weird looking enzyme. It sort of looks like a donut or a bagel. It's got a hole in the middle, and one of the ends of one of the strands of DNA threads through that hole, and the thing just sort of travels down the DNA strand and unzips it. It starts at something called a replication fork, which is not at the end of the DNA, but could be anywhere in the middle. Remember, we look for a place where it's easy to separate the strands. That would be places where there are lots of A's and T's. Once the helicase is attached, one strand goes through the hole, the helicase unzips the molecule, and it travels down the strand at about 300 nucleotides per second. You can do a little math and figure out if it's 300 nucleotides per second and 3 billion base pairs in length for one molecule of DNA, it's still going to take a tremendously long time for one helicase molecule to unzip the whole DNA molecule. So you've got thousands and thousands of them all along the length, unzipping a little portion of it, getting it ready to be copied. Once you have the DNA open and the bases exposed, you're going to use an enzyme called primase. There's the ASE ending. Primase builds a primer. Now the weird thing about DNA is that DNA can't copy itself. It can't start a new chain by itself. You need a primer, like a seed. And the seed has to be made out of RNA. It's a very strange situation, but so the primer itself is made out of RNA nucleotides. At the end, we got to get rid of that because we can only have DNA nucleotides in our DNA. So we'll get rid of that later. Okay? Now that we've got the primer in place, the most important enzyme, I think, in this process comes in, and that's DNA polymerase, DNAP for short. DNA polymerase, again, ends in ASE, but its first part of its name is DNA polymer. So DNA polymerase acts on or makes the polymer. It brings in DNA nucleotides, it matches them up to the exposed bases on this in the replication fork. This will happen on the template strand first and then on the other strand. And as it synthesizes, this is the really neat thing about DNA polymerase, as it synthesizes a new strand of DNA, it proofreads it, looking for mistakes, removing mismatched bases, fixing any errors that it encounters as it's going. It's going to move pretty fast. And again, it's happening all over the DNA strand, tons of them at the same time. Finally, we have to kind of finish the process because all we've got are these DNA nucleotides just sort of attached or attracted to the exposed bases. So the first thing we need to do is get rid of the RNA primer, so we use exonuclease. And then finally we need to seal the sugar phosphate backbone to make a complete strand, and that's done by an enzyme called ligase. Again, the ASE. Lig, L-I-G, comes from a Latin word to tie. And so what ligase does is it ties together the nucleotides. Now remember, these strands are directional. They have a 3' end and a 5' end, and DNA replication only goes in one direction on the strand. It's the new nucleotides are added to the 3' end the growing DNA strand, which means that the replication is proceeding from the 5' end to the 3' end. That's really important because remember that the strands are oriented antiparallel, which means that as DNA polymerase is trying to travel down one strand, on the other strand it has to go the other way. Well, that's problematic because the fork, the replication fork, only opens in one direction. And so on one of the strands, DNA polymerase is able to just travel down nice and smoothly right along with the helicase and just synthesize the new DNA as the fork is opening. But on the other strand, we have some problems because we have to synthesize in the opposite direction. And so that means we have to wait for the fork to open a little bit, and then we have to synthesize it backwards, and then we have to wait for it to open a little bit more, and then synthesize it backwards. That's called discontinuous replication. And it's going to cause some problems because that means it's not going to be as fast. On the one strand where the DNA polymerase can go from 5' to 3', that's called the leading strand, and it's nice and smooth, no problems, nice, easy synthesis. But on the other strand, because it's going in the opposite direction, the replication is discontinuous, and we call that the lagging strand. Lag means to sort of hang behind, and the lagging strand takes longer. It's also where you're going to have the most errors. So on the lagging strand, because we have to do it in these little choppy fragments, those are called Okazaki fragments, which is named after the Japanese scientist who discovered them. Most of the errors are going to happen in this choppy section. Here's a picture to show you what it looks like. So at the very top you can see the replication fork is opening, and in the way it's drawn here, it's opening from left to right. So as that little red arrow is pointing to the right, that's the direction we're opening up the DNA. The bottom strand is 5' to 3' from left to right, and so the DNA polymerase is able to make new DNA along that strand as it's following the replication fork. The top strand, though, is 3' to 5'. It's backwards. It's antiparallel. And so you'll notice the little black arrow, that's a primer. We have to put a primer in place, and we can only synthesize back towards the left. So we've made a little piece of DNA going back towards the left, but now the replication fork has opened even further. So we need to put a new primer in, and we need to make a new piece of DNA. And then we have to wait for it to open up a little bit more, and we have to put a new primer in and make another piece of DNA. That choppy, discontinuous replication is where your errors are going to happen on the lagging strand. So one of your two copies of this molecule will come out nice and perfect, and the other one is going to have some errors in it. Fortunately, there are ways of repairing those errors that we'll get into later.